My name is Begoña. I am from Spain, where I have studied my master's degree in chemical engineering. I came here and I am doing my second master in food safety, hygiene and management. The reaction of browning is oxidation of polyphenols that the apples contain and is catalyzed by the oxygen. But we could say also that there is a mass transfer since we are dipping the apple slices in the formulation. So there is a mass transfer of the main chemicals from the fluid where we are dipping to the surface of the apple. Basically, we get apples from supermarket, we wash them, we peel them, and after we dip in the formulation that we have already prepared before. We dip for 5 minutes. We introduce the apple slices in plastic bags that we seal, and sometimes we vacuum before seal to see the difference between how the reaction is different with and without oxygen, since the oxygen is the main catalyzer of this reaction. And we measure the color with an instrument called colorimeter. For time zero, after the samples goes to the fridge or to an incubator to 25 degrees, and we are monitoring these samples about 24 hours after, 2 days after, 3 days after, 14 days after, and 21 days after. The samples that we put in the fridge are in the same conditions that we can find in the supermarket. And the reason why we put samples also in the incubator at 25 degrees is to see the accelerated reaction, because at 25 degrees the apples will get brown quicker. So we are using different formulations. We are using water to compare the white of the experiments to compare our formulations, and we are using sulfites also to compare, because the basic is our aim is that our formulation work as well as the sulfites do. So we have to compare with sulfites to see if the formulation is good enough or not.
My name is Biochemical. I did a BEng in chemical engineering in University of Birmingham, and I moved over to Birmingham to do a master's also in chemical engineering. Sulfites are banned nowadays in the food industry, so I'm trying to look for alternative to sulfites. They help preserve drinks and juice and apples and oranges, so I'm looking for alternatives for sulfites. Also, we'll peel them, wash them and blend them. And then we put them in the centrifuge to remove the supernatant. That is what we use to take our samples. So we pasteurize them to get rid of any bacteria there might be. We mix our juice with the chemicals. I've got a lot of samples, 18 samples. So I've got samples with the juice and water, samples with the juice and sulfites, and other alternatives I want to use, like polymer as well, just a bunch of different samples. So we mix that. We store them in 3 different temperatures: 5 degrees, room temperature at 50. And every day we take the samples out, take a specific volume to make measurements. We're looking for pH, we're monitoring pH, how much it browns, we're looking for absorbance, and we're looking at how much vitamin C degrades. So every day we take those 5 samples from the actual sample, and put it back in the fridge after, and just keep doing that on a regular basis and see how much it browns. This research is mostly for the food industry, but obviously as chemical engineers we can we are useful and work in the food industry. And the reason why I'm doing this is because just for the experience, the diversity of it, and maybe someday I might decide to work in the food industry, and this would be very useful that I've had this experience on my CV. So